After her presentation, Maria Pilar Velasco that is a PhD student working with us here at the Institute for Sustainable Agriculture, will present some of the very recent results. We just got some results yesterday in the afternoon that we are obtaining to characterize how similar is this strain with the Italian ST53 strain from Puglia region. So Marta, when you want, please,
okay.
the time.
Okay, so I will start to sharing. Do you see well the presentation?
Yes, no.
Yes.
It's okay?
Yes, perfect.
Okay. You hear me well also?
Yes.
Yes, perfect.
Okay. Okay. So hello to everyone. As Blanca have said, I'm the head of plant health in Balearic Islands, and I will do a sorry, but the screen is not following. I will. So you see it well. Sorry. Yeah. I was in the last one. Now you see well. Okay. Yes. So it's going to be a brief presentation. I will first update the current status of the bacteria in Balearic Island, and after we will see a photographic annex of the latest developments of the harmful organism in specific in the new focus of pauca. As you may know, on October 6th of 2016, the official laboratory of plant health of the Balearic Islands detected the bacteria in a cherry tree at a garden center in Mallorca. This marked the first sighting in Balearic Islands, but also in Spain, of this harmful organism. Subsequent positive emerged not only in Mallorca, but also in Menorca and Ibiza, and the entirety of Balearic Island is encompassed within a single infected zone as established by the Commission Implementing Regulation. On August 3, it became our decree 65/2019 that was published, declaring the fight against Xylella as a matter of public interest in the autonomous community, along with establishing mandatory phytosanitary measures to combat this plague. As you can see here, the distribution spans across the three mentioned islands. You can see that Formentera is free by the moment, and these multiple genotypes that we have here in the islands, it indicates that we have multiple introductions, and during a lot of years, in extensive of years. Among the numerous actions taken to prevent the bacteria spread, it was the immediate prohibition to export specified plants from the Balearic Islands outside, but we also have prohibitions in the movements between the islands. These are the cumulative data that we have since we started we detected Xylella till 11 of March. That is the last official notification that we have done. We have taken more than 21,000 samples, of samples of vegetal material. This almost the part of all of these samples have been analyzed, and we have 1,073, I don't see the screen, but 1,073 positives in all the islands. Here you have also all the numbers between every island. So I have said the number of species prospected is 487, and the number of species that have been detected positive in Balearic Islands are 38. These species are these ones that are here. There are a lot of species, and some of them important that have agricultural importance here in the islands, like olive trees, like Prunus dulcis, and also importance for the Mediterranean region, like Salvia rosmarinus, lavandas, all these species. Maintaining a continuous surveillance and adopting preventive measures to mitigate the impact of this bacteria on agriculture and local biodiversity is essential, and the collaboration between institutions and active community participation are crucial to addressing this challenge. We have an action plan that is updated annually and every time that is needed, like in this case, that we have updated with this special situation with the new subspecies. And we have a statistical basic surveys that are conducted using the Rivers tool. In the vicinity of locations with a particular cultural and social value, we take more samples for protect these areas, and also we do surveys in the 50 meters of every positive finding. We do information campaigns. We do provision of phytosanitary products for the vector control in agriculture areas, and the prohibition of planting certain non-resistant species and varieties are one of part of our measures. So this was the background, and now we are starting to the point of the presentation. The 5th of January of this last January, the first positive of Xylella subspecies pauca ST53 was identified in a symptomatic wild olive in the municipality of Sencelles, in the island of Mallorca, in the center of Mallorca. This discovery marks a new milestone in the phytosanitary surveillance of the region. All started with a citizen alert that her olive plantation was in a very poor condition. The same week that we received this alert, a plant health technician visited the plantation and found symptoms of Xylella in the olive trees. But additionally, he noticed that some wild olive trees on the edge of the plot were in very poor condition. So samples were taken from both olives, the olive plantation and also the wild olive, that which tested positive for Xylella in our laboratory. But this wild olive was also sent to Blanca's laboratory in Córdoba for further analysis. That reveals that we have a new positive for pauca ST53. That was a very surprising fact to everyone. So currently we have 3 subspecies in the Balearic Island. We have Xylella fastidiosa multiplex ST81 in Mallorca, Xylella fastidiosa pauca ST80 in Ibiza. I have mentioned that Formentera continues to be free of the subspecies. And in Mallorca, we collect all of them, it seems that we are happy to have a lot of subspecies. And we have subspecies in red is multiplex ST81 and also ST7. And so the green one is Xylella fastidiosa ST1. And the new one, that is this purple point, that is Xylella fastidiosa pauca, is the 53. This is the focus of pauca. It's a 500 meters radius, and the blue line is the 1,000 radius meters. So we have taken, sorry, but I don't see the number. I think it's around 600 of samples. Of these samples, 153 have been positive, and we have these negative samples, and we have about 100 samples that are pending to analyze. The samples that are being more positives are the wild oleas and also olive trees, but we find it also in Rosmarinus officinalis and in Rhamnus alaternus. These are the positives for Xylella, and in this slide you can see the ones that have been positive for multiples, pauca, fastidiosa, and we have also detected some co-infections of these two subspecies. You can see that we have positive for pauca for all white oleas and also for olive trees, but we have some positives in a ornamental plant, it's oleander, and Rhamnus alaternus also. This is the radius also again of the pauca. This is the position of the three positives by subspecies fastidiosa. We only have these three. These are for the multiplex variety, and these are the pauca positives. You can see that we have a very big concentration here in this municipality. This slide I put it here because it reveals the area where is affected this pauca. You can see that it's an agricultural area, but there is also an urban area because there are some houses here, and also some of forested and shrubland zones intersect. This make challenging to identify every plot, to identify the owners of every plot, and to do a proper survey and a proper elimination of every positive. And if you can see here, this is the plantation of the olive trees that the citizen alerts on us in the beginning of January. So this is like, I think that you can't read anything, but I just put it here because you can see that these are the samples and the resamples that we take in this plot in the olive plantation of this citizen, and we detected 50 positives in just this small plantation of olive trees from 67 samples. So you can have an idea of the quantity of samples that we have taken and the status of the bacteria in that zone. Here, these slides showcase the species that have been positive two different subspecies of Xylella fastidiosa. These ones are for multiplex ST81, that it's the one that we have more species detected, and also for multiplex ST7. This is for Mallorca. This is for fastidiosa ST1, and we have all of these species, and also for Xylella subspecies pauca. This slide is not being updated because in the last week before Easter, we have the confirmation of the CSIC laboratory of Blanca that all of these species that have been determined for pauca are ST53. So we have the confirmation that in Mallorca all the ST that we have here are 53. And here are the subspecies for Ibiza, for subspecies pauca ST80. And these are the subspecies multiplex ST81 that affect in Menorca. So since 2017, we have taken a lot of samples, and 405 samples were extracted, and we determined the ST that we have it, and all was multiplex. But it was pending 167 at the end samples positive from olea, wild oleas, and also olive trees. 106 were from Mallorca, 61 were samples from Menorca, and all of these samples that were old samples, not now, from all Mallorca and from all Menorca, we determined that 13 olive trees were multiplex, 19 wild olive trees were multiplex also, and wild olive tree was fastidiosa that we did. This was a new fact, because in the past we don't have wild olive trees affected by fastidiosa. And in Menorca, all the wild olives were multiplex. So this indicates us that the outbreak of subspecies pauca ST53 on the island of Mallorca seems to be confined to the vicinity of the first positive in that area that I have shown you in one moment. Yeah. And now we will start to see all the photos that we have done in that area, and you can see how is the affected area. This is all wild olive tree near the municipality recycling point that you can see that it's completely defoliated.
Wow. Oh gosh.
Another wild tree that appears completely defoliated also. This is the symptoms that we have seen. It are symptoms of general Xylella, but we haven't seen in Mallorca that wild olive trees so with this clear affection and so bad. This is a 100-year olive tree that has tested positive for a co-infection of Xylella multiplex and pauca. Nerium oleanders affected by Xylella in the vicinity of the ministry of the municipality of Sencelles. Another Nerium oleander that is completely defoliated. This is an Nerium oleander that despite this, its symptoms has not tested positive for Xylella. olive trees showing symptoms. You can see all the photos. These are the olive trees of the plantation of this citizen that alert us of the poor conditions. More symptoms of olive trees of the plantation. Again, the plantation of the olive trees. You can see that the trees have been pruned a lot, and there is also some symptoms of defoliation. A wild olive tree with symptoms, and this is one of the eliminations that we have done in the zone, in the affected zone of pauca. And we covered to avoid the regrowth of the wild olives, and after that all the reds are burned in a near plot. And I think that it's all. I don't know if you have questions. Of course, you can do the questions that you want, and if you don't have questions now, but in the future, you have here our mail, Sanitat Vegetal, that you can write us to ask for anything that you need. So thank you so much for your attention, and thank you, Blanca, to give me the opportunity to speak about the pauca in Mallorca this afternoon.
Thank you, Marta. I think we will continue with Pilar, and then at the end we can wait for the questions of the people that can be writing in the chat. Okay, so Pilar, the floor is yours.
Perfect.
So could you see my screen?
Yes.
Okay. So as Blanca said before, I'm Pilar Velasco. I'm a PhD student at the Institute for Sustainable Agriculture in the Crop Protection Department. And as an objective of my PhD, we develop a next generation sequencing tool for the massive study of the genetic diversity of Xylella fastidiosa population and its main subspecies. And we recently use it for study the new ST53 that we have in Mallorca. Okay, so as many as you know, so there is an Commission implementing regulation that and there is specified a list of tests for the identification of Xylella fastidiosa, and here in Spain for the surveillance, tracking and diagnosis program. The first step is the sampling. So it's carried out by the autonomous communities in the plant health laboratory in Balearic Island and also in the plant health laboratory in the Valencian community. When these laboratories has a positive sample, then they send the sample to the National Reference Laboratory for Phytopathogenic Bacteria to confirm the diagnosis. And at the end, they send to us the sample to characterize to the subspecies level. So in this regulation, there is many tests that you can use. For instance, for the identification of the presence of Xylella fastidiosa, you can use different protocols as qPCR, the loop-mediated isothermal amplification, and also conventional PCR, and also for the molecular test for the identification of the subspecies, that it can be used the MLST and also other conventional PCR to determine the different subspecies. these two protocols are not included in this regulation, but are included in the EPO protocol, and also discriminate between the subspecies. So the step that we follow normally is when we receive a positive sample infected by Xylella fastidiosa, we perform the MLST analysis, and if it's positive, we know the ST and also the subspecies. And if we get no amplification, we normally perform the nested MLST or the duplex or tetraplex qPCR, that we normally have the subspecies that are infected this sample. When we carry out the sampling that we normally perform, we realize that we get the genes or the different locus from the ST53. So we start to repeat all the MLST analysis, and to confirm that we have the ST53 that was the subspecies pauca and was a new ST that was not described in Mallorca. And Marta said more updated information of this, but to the date we have different host plant that was infected by this ST53. So going back again to the regulation, we have to say that most of these protocol work well and was optimized for Xylella fastidiosa, to work with Xylella fastidiosa culture, but these protocols are more problematic with plant DNA because due to the plant inhibitor. And also, there is another inconvenience with the MLST analysis, that there is a lack of monophily for isolate assigned to the subspecies multiplex XT6 by the MLST analysis between the XT6 from Alicante and from Corsica. So the best option were to have a complete assembling genomes that could reveal novel features such as the plasmid, the phage, and genome reorganization that also could not be detected otherwise. But we have to say that we are talking about Xylella fastidiosa that has a slow growth and also has a very specific nutritional requirements depending on the subspecies. So the isolation of the bacteria is not a good option. So there is a need for a fast and accurate approach for the Xylella fastidiosa typing. So these two scientific papers are based on the next generation sequencing that is a powerful method with facilitate a rapid deep sequencing of the DNA sample. But the problem, again, is that within the whole plant genomes, the actual sequence region relevant for Xylella fastidiosa diagnosis are typically at low relative abundance. So we would require a deep shotgun sequencing to accurately detect and reconstruct them to extract as much as possible information for the correct diagnosis. So this is the step for the protocol that we develop, is that when we have a positive Xylella fastidiosa samples, doesn't matter if it's plant or insect, we make indexes library, and with bytes, we perform a hybridization. So these bytes is going to be attached in the Xylella fastidiosa DNA sequencing, and then we cleaning this sequence. So we are going only to return the Xylella fastidiosa DNA, And after all of them, we are going to have an enrichment capture library that could be sequenced in any of these platforms and also BackBio. Another thing that is really good of this protocol is that we can enrich multiple problem samples simultaneously at the same time. So this decreases also the cost of this process. So we selected 137 target sequencing that cover conserved and no conserved region of the different subspecies and sequence type known of Xylella fastidiosa strain. And this target sequencing correspond to the MLST, also genes that differentiate between certain subspecies, also the MLSA that are environmentally mediated genes. The most important that we have 107 genes that correspond to the pangenomes, and also because of the importance of the different plasmid profile that we have here in Spain, we decide to design also some bytes that go to the plasmid. And we designed 7,095 oligonucleotides bits, and finally, after an screening to see if goes well, we finally retain 7,165 oligonucleotides. So after the sequencing, we have to do a in silico bioinformatic analysis. So the raw reads of Illumina, we have to trimming. We perform a quality filtering, the adapter removal. We also, with Kraken, we assign the taxonomical levels of this short DNA sequence. And we extract the reads classified as Xylella fastidiosa. We perform a sequence assembling and also an annotation, and finally we can do the typing of this MLST. So here you can see the sample from Mallorca, the ST53, that you can see that after the library, to perform the library and sequence the sample, all the percentage of the reads are classified as Oleoeuropea, and only an 8% of these reads were classified as bacteria, and also a 2% of Proteobacteria. But after this capture procedure, you can see that the percentage of reads classified as Oleoeuropea, as plants, is lowest, and we have a lot of reads classified as Xylella fastidiosa. So we were able to capture all the genes for the MLST, but we also could capture a total of 133 genes from a total of 140 genes that was the target genes. And we also confirm that with this method, we also have exactly the same allelic profile that we obtain with the normal MLST. And with the total of all the genes that were reconstructed, we could perform a phylogenetic analysis. And here you can see that this sample, the Senceles olive sample, that is in the middle between the ST53 from Italy and also the ST53 from Costa Rica. I have to say that here we have also another sample that we perform this sample was from Argentina, that was this sample show a different allelic profile, share some alleles from the subspecies fastidiosa and another from the subspecies pauca. So we decide also to perform this protocol, this capture protocol, and you can see here that is in the middle between pauca and temecula. And I have to say that this result I get between yesterday night and this morning is there are really fresh result. And nothing, I would like to thank to my lab and all the projects that are involved in these protocols. And if you have any question, I'd be happy to answer you.
Thank you, Pilar. So now for those that are connected, please, if you want to make question, write in the chat.